In this video, we will discuss about this PDAC tumor microenvironment remodeling in order to diagnose the patients who are affected with this pancreatic cancer. So these are the pancreatic cancers which are represented by this green color cells, and usually there is a stroma surrounding this pancreatic cancer and also the tumor microenvironment, and both together act as a mechanical barriers that can prevent the immune response against the pancreatic cancer cells. And in order to rapidly grow and proliferate, cancer cells need to be in the glycolytic state, and in this glycolytic state they need more, or they consume more glutamine, and because of that what will happen is that the cancer cells will upregulate this FAK signaling pathways, and these FAK signaling pathways can also be triggered by the TGF beta in the tumor microenvironment, and both in together helps the pancreatic cancer cells towards proliferate and migrate, or towards proliferations and migrations. And how this tumor microenvironment remodeling can be done in a way that it can help in the immune response, or increase the immune response efficiency against the pancreatic cancer cells. One of the studies has been done where they inhibit this glutamine. By inhibiting the glutamine, they will allow these T cells to come and interact with this pancreatic cancer cells, as the glutamine will inhibit the immune cell activity or its activations, and thereby it will help in the increasing the efficiencies of the immune system against the pancreatic cancer cells. And along with this glutamine, if there has also been studies which has done to also inhibit this PD1 receptors through the anti-PD1 ligand, or we can say the immune checkpoint blockade approach. So when both of them are combined, that is glutamine inhibitions and the ICV approach, then they will have a better results compared to the case where individual therapies are being applied. And by inhibiting this glutamine, this hyaluronan synthesis will also be reduced, and this will reduce the ECM productions of fibrosis. That is one of the main component to act as a mechanical barriers that prevents the immune systems from detecting these pancreatic cancer cells, or prevent the immune invasiveness. So by inhibiting this glutamine, one study also shows that hyaluronan synthesis will also goes down, thereby the ECM of fibrosis will goes down, and the FAK, that is focal adhesion kinase signaling pathways, will also suppressed, and thereby the tumor microenvironment remodeling will occur, as it will also have an impact on this TGF beta, and this in all turns will weaken the stroma that has the anti-tumor properties surrounding the pancreatic cancer cells, and helps the immune systems to invade this stroma and detect the pancreatic cancer cells and generate the antibodies that is specific to the pancreatic cancer cells. The other things is about this macrophages and MDSC. So macrophages are, as we know that there are two populations of macrophages, M1 macrophages and M2 macrophages. These M2 type macrophages, which has a tumor proliferation properties, are usually found in the stroma region in the pancreatic, surrounding the pancreatic cancer cells, or in the tumor microenvironment of the pancreatic cancer cells. So these things will also be suppressed once the stroma gets disturbed, and the differentiation of this myeloids into the M1 type macrophages will be more compared to the M2 type macrophages. So this is some of the basic overview of how this PDAC remodeling can be done either by this glutamine inhibitions and which can be combined with the ICV therapies to have a maximum impact of the diagnosis on the pancreatic cancer cells. That's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.